to lecture 18 of the Learning About Learning lecture series. So far we have been looking at and dissecting out the molecular basis for the complex behaviors. More specifically, in the last lecture we were talking about the role of NMDA receptors. NMDA receptors, these are a class of a subclass of glutamatergic receptors, and we were looking at what effect do they have when on LTP when they are being taken away from when the gene that is responsible for generating these receptors are taken away from an animal. Specifically this taking it out or the knockout is specifically localized to a region of the brain called the hippocampus, and even the sub region of the hippocampus CA1. The localization is so good we can actually go down and then dissect out the molecule only in the CA1 region, right. How do we know that? We saw that this picture, and where we said, hey look, you can go ahead and stain for the presence or absence of this molecule in terms of its mRNA, right. It is a one, these receptors are proteins, and proteins are expressed in a living system by translating from translation from mRNA. So, you are actually looking at the mRNA stain here, and then you can see that that stain is missing specifically in the CA1 region. Just I want to point out one little thing here. This is how deep and how good a control we have in terms of asking the questions such as what role a molecule in a region plays as compared to Carl Lashley, where he was taking out chunks of brain. See, the motive of the questions are more or less the same here, right. You want to know the substrate of where the memory could be. You want to know what are all the responsible factors, or what are all the factors that are responsible for forming the memory. From the days of removing chunks of brain, we have gone to the place where you can actually go down to a region of the brain, in fact a sub region of the brain identified by the differences in the cell type of the neurons that are present there. In that region specifically go ahead and remove R gene and nothing else. Now that causes a deficit in LTP, long time potentiation. We were wondering what this could mean in terms of the animal's behavior as a whole. How do you probe this behavior? We probe this behavior through several means, and one of the behavior that we were looking at is a water maze experiment. And where you can where we were describing how the experiment was done. So, briefly the experiment is done by placing the mice in a pool of water and asking it to find a platform that is hidden inside that pool. Now, over a period of time you will see that the time required for the mice to find, or any rodent, for mice or the rat to find the platform will go down, but more importantly if you actually section out or divide that entire pole into 4 little quadrants and then ask where the mice is searching predominantly when you remove a platform. This is a probe trial, right, when you remove a platform, and now you are asking where is the mice searching now predominantly. You see that they are searching in the quadrant where the platform is usually present. Now, the definition of these quadrants comes from the fact that there is a platform, and then the mice know this is where the platform is, because not because it can see the platform, but it is actually computing where the platform could be based on the cues that are present outside of the maze. So, it needs to make two things here. One, it needs to understand what are all the cues that are present outside of the maze, it is called the extra maze cues. It needs to remember that in relation to those cues where the platform is. Now, that is exactly what you are testing, and that is the memory here. And we said this is also dependent on hippocampus. If you lesion out the hippocampus in a regular training, the mice fails to figure out where this platform is. Of course, you can overtrain them and then make them learn that it is make them learn to search in that particular quadrant, but the point is in regular training they would need a hippocampus, and if you lesion out them, if you lesion out the hippocampus, they would not be able to locally search this quadrant. Now, what happens to these mice in whose CA1 region lacks NMDA receptor? Now, can we actually take these mice and then put them to this behavior protocol, and then if you do that, they clearly learn, you see that latency coming down, but the important point that as I was stressing is that you want to test their memory, right. A memory is tested when you do not in this particular behavior, when you do not have a CS equivalent of a signal here, right. You remove the platform and you are asking the mice to actually figure out, sorry, there is no US present there. So, you remove the platform and then you are asking the mice to probe, probing the mice, can it actually figure out where is the platform would be. When you do that, that is what you see. What we are plotting here is the search time, right, in the probe trial, okay, and then we are asking how much of the time the mice is actually spending in each of the quadrants. Okay. I want to draw your attention to the black bars here. They represent the knockout animals, similar to the LTP measurements, where we had a few bunch of controls, which I said do not have to worry about, we just concentrate on the meat of this experiment, where the bunch of controls are done to ensure that we are probing the right thing, right. So, you can think of them all, meaning you can take a regular control, that is the in this case it is the open bar, and compare to the open bar here, you can see these mice, the knockout animals, do not search in the target quadrant at all. In fact, they search in all the quadrants almost equal with equal probability, just at the chance level, compared to the controls where they specifically search on the target quadrant. This is significantly more compared to the rest of the quadrants, and that is again a point or a proof that these animals, when you knock them off of NMDA receptors, specifically in CA1 region, is unable to form a memory of where the platform is located, right. You can do some quite neat variations of this experiment here, where you remember the water maze and the platform. What you can actually do is you can place a visual cue like a post, like a flag post there. You will see they beautifully learn to go navigate to that flag post and then launch out to the platform. So, there is no problem of understanding how to reach there, that is not the problem here. They simply do not know where is the platform. Now, that is very convincing that you need these coincident detectors, these receptors that can get activated only in response to both the pre and the post neurons activation. Those you need these receptors to form this associative memory, and we also know that they are required for forming the LTP, long term potentiation in this region. Now, this is all about forming or inducing the potentiation, right. Now, I have described to you the function of these receptors. Clearly, from just by this function, there is no simple way for you to extrapolate to the fact how, after removing the stimulus and once that is gone forever, how can they maintain their higher level of throughput, right. The memory is not just about changing the throughput, but being able to maintain that over a period of time, right. That is same is true with LTP too, right. It is not just sufficient to induce it, induction is not just similar to the memory, it is induction together with the maintenance, you need both of them. Now, how do you I think that is that can that be done at a different level, or can that be done by another molecule? Now, that is a big question, because whenever it comes to maintaining a molecule or a memory through a molecule, then you are talking about maintaining over time scales that are larger than the molecular turnover time scales. Okay. Typically, let us say if a memory were to be encoded by a molecule A, you can say, okay, I am going to increase the levels of the molecule A, and that molecule, then that level of that molecule should probably encode that memory. However, the problem is each and every molecule in our body, in a living system, has a finite lifetime. What happens to this memory after this lifetime? It is not infinite, just like our lifetime is much longer than this molecule's lifetime. So, they do get turnover much faster, much faster in fact compared to our anybody's longevity here. So, to solve this seemingly contradicting problem, so one thing to say is, okay, maybe the memories are not represented in the molecules, but somewhere else, or to come up with an real proposition, real idea how the molecules can act like a switch, almost to say you turn on, event turns on the switch and it maintains that state until something else happens. Okay. How can you do that without being susceptible to this molecular turnover? In 1995, John Lissman proposed this, came up with this unique idea, by which he said, hey, we know in biology many of these enzymes go from activated to an inactivated state through a signaling mechanism called phosphorylation. This is addition of a phosphate group to the enzyme or a molecule by a living system. Now, if that were to be possible, then maybe something related to that can be utilized in here to store the memory too. Again, the main problem is the mechanism that we are going to propose need to be robust enough and need to be overcoming the problems faced by the turnover. So, it cannot be just the phosphorylation, because the phosphorylated enzymes are at some point going to be decomposed and going to be taken care of by the living system. So, it can't be simple phosphorylation. If it can't be simple phosphorylation, then what else could it be? He said there can be a generic mechanism, phosphorylation being one example, but a generic mechanism wherein there is activity that is turning from one state to the other, like the phosphorylated to the unphosphorylated state. That is very much same as what we have been talking about. Okay. Now, let us look at this proposition here, where he says you have a molecule K1 and the molecule K1 gets phosphorylated to become active molecule K star. Now, in biology, in living systems, we have both these reactions possible, that is forward reaction where this molecule gets phosphorylated through addition of a phosphate group and becoming activated molecule, and they can also be dephosphorylated by a set of enzymes called phosphatases. There are kinases which adds the phosphate to make the molecules activated, then there is a set of enzymes that can take away this phosphate and then revert it back to the original state. If you were to hypothesize that the activated molecule are responsible for neuronal function, number one assumption of the hypothesis, it is the activated molecule that are responsible for neuronal function, like maintaining the plasticity and so on and so forth. Second, the crucial point being the activated molecules themselves can self catalyze their own phosphorylation, that is the key here. If you can these molecules themselves, right. So, can act as their own enzymes, their own kinases, such that this molecule becomes active, they can actually in turn activate more of their fellow beings and so on and so forth. It can be an infinite cycle, because there are also reverse reaction possible, the phosphatase is possible, right. So, in such a system you will first achieve an equilibrium, but that equilibrium can be affected by arrival of a stimulus, the S is hiding here. So, stimulus that stimulus, and the stimulus when it comes through some other kinase or some other mechanism, they momentarily increase the kinase activity momentarily, that is the key here. That increase is sufficient to flip this whole equilibrium from one state to the other. Okay. Now, this is all good and saying it in words, but the beauty of this is that it is such a simple or reasonably simple chemical reaction. You can go ahead and write down the rate equations and the concentrations of K1, K1 activated P as a function of time and see how is the behavior of the system. So, what is it I am looking for? What I am looking for is, let us say, the fraction of the molecules, the total molecules, the K here we are talking about, since it can add phosphate group on its own, they are the kinases, we are looking at kinase class of compounds which can self auto phosphorylate itself, and we are looking at specifically the concentration of the activated kinase, the fraction of the activated kinase, and how it is changing or how that concentration affects the subsequent rate of formation or deformation. Why I am interested in this subsequent rate of formation or deformation? What I want to know is that I want to look at stable states. What do you mean by stable states? When we are when you are plotting these rates as a function of this K1, the activated state concentration, I want to look at a place or multiple places where the concentration of the activated state does not change. It takes a lot of pushing or pulling to flip from one level of concentration to the other. And it turns out you can actually go ahead and calculate, write down these equations and calculate this particular rate. The rate here we are talking about is the rate of decomposition or rate of change of K1 activated state as a function of time. So, that is what we are actually plotting here as a function of the fractional, this is actually K, capital K activated estimated as a fraction of the total K. All right. Now, what I am looking for here? So, now let us look at few of the points here, a point 0 here and then a point here, right about here. They are characterized by these stable states. Why I am saying that? If if the concentration were to increase around this point a little bit, then what is going to happen is the rate is negative and it increases really fast here. This negative increase in the rate, which is the deacceleration so to speak, will pull down the concentration back to 0, because you are actually reducing the rate at which the K1 activated state is forming, and the phosphatase is good enough to bring it back to 0. Same is the case here. Instead of now bringing it to 0, it brings to somewhere in between, it is about 0.75 if you 0.74 if you actually go back to this paper and look at the number. So, except for these 2 points, nowhere else, this is not a good point, because depending on whether you are high on this side, I mean you are deviating from this side or that side, it is actually a rising slope, right. So, it is going to this is going to pull it forward, and that mean it is a reduction in the negativity, and as a result the whole slope is positive. We are actually looking for a negative slope around that point, so that it can actually pull it back. So, these two states actually form a very good stable states, and in fact now you can go ahead and ask, if they are forming these stable states, can I simulate a stimulus arriving and then monitor or plot the concentration of the k star p 1 p as a function of time when the stimulus is arriving, and look at that behavior. And they did that in that paper, and you can look at this now, where we are plotting as a function of time two things. Number one, the stimulus, the K2 in terms of the activated K2 here, that is in terms of nanomolars, does not matter, is the concentration of the stimulus. So, you are giving that stimulus and clearly the system is responding, the fraction of the K1 activated state is going up a little bit until a certain point right about here, where it completely goes up and then stays there. Now, that is memory. It went from this state to that state. Now, no longer you are giving the stimulus and it is continuing to maintain it there. You have flipped the switch from one state to the other state, all that by identifying a way in which there is a self catalysis of a reaction of an activator, so to speak, and that is taking care of much of your neuronal function. That is all we are told. Of course, implicit in this entire assumption is this the reverse reaction is catalyzed by some other enzyme, and that is happening all by itself. There is now there are interesting reasons why this would the system would behave the way it is, but that is purely enzyme kinetics and really chemical kinetics there. And but the bottom line message that I want to drive is that we can, if we want to look for a molecule, we want to look for a molecule that needs to have self activation or self catalysis, and that need to be some kind of a kinase equivalent of that molecule, and that can have a function. Why I am talking about the kinase here? Because it needs to have a ability to modify the neuronal function. It needs to activate few other molecules down there which can actually carry forward the function which increases the neuronal conductivity or the efficacy. People looked for those kind of molecules, and one potential candidate happened to be alpha Chem K2, abbreviated as alpha Chem K2. It is also called alpha calcium calmolidin dependent kinase 2. Alpha is one form of this big protein, and they hypothesized that the Lisman again they hypothesized that this could be a potential target for maintaining the memory. So, alpha Chem K2, the calcium dependent calmolidin kinase 2, what does it do? Number one, we need to make sure that we are able to respond to the NMDAR signal, right, the NMDA receptor signaling, right. We have opened, NMDA receptors act like coincident detectors, they have opened up and then the calcium flowing in. So, now first thing you want to make sure is that alpha Chem K2, this enzyme, is physically present in a place where the NMDR signaling mechanism is happening, and you can actually detect this enzymes need to be able to respond to that. And the first thing that they saw was that you can actually they are the alpha Chem K2, you can actually do a similar localization study like the staining that we saw in the NMDR knockout case. We can actually do that, and then when you do that you will see that they are highly enriched in the synaptic spines, dendritic spines. These are the these are the regions of the postsynaptic neuron through which the postsynaptic neuron receives the information, they are this is where the NMDA are localized to. And what we want to see is that when we are doing the NMDAR based signaling, they are localized to the synapses, I mean the dendritic spines, and that is what has been shown here. So, what they have done is they have taken this neuron and loaded this neuron with the calcium dye, and they went there near the spine and then locally uncaged through a photochemical process. You can release a bunch of glutamate molecules. These are these are compounds which form through a chemical bond cage the glutamate. I mean they do not let them move free. So, they connect them and then keep it there, but when you send in a photoactive pulse, which is basically a light pulse, and when you do that, that bond gets cleaved and then the glutamate is now free to move. You need to do that because you want to get that spatial localization, so that you can go to the spine and then release just there and not anywhere else. And when you do that, what they see is that this stimulus potentiation causing stimulus is releasing when it is released near the spine, it is activating the spine, and it is dependent on NMDA receptors. How do we know it? Because you can use a drug AP5 and that blocks the NMDA receptor, and you do not see this calcium signal for this stimuli anymore. So, clearly saying that the NMDA receptors are localized in spines through other staining. We know that the Chem K2 are also localized in the spines. Now, physically they are in the same place, so that problem is solved, but does our alpha Chem K2 qualify the criteria that we have posed, that is posed by John Listman? In order to answer that question, let us take a closer look at the molecule itself and its structure. When you look at that, so it is a protein, it has multiple domains or multiple structural elements, each doing its own function. There is a large portion of a catalytic domain, and there is also a calmolidin binding or an inhibitory domain, and so on and so forth. Okay. So, now if you actually that is when you are actually lining up all the constituents of this protein and then lining them up and then asking what these functions are. But if you actually look at the structure, you can kind of approximate, or you can kind of draw this hairpin like structure to them. What you see is that there are specific areas, and these specific areas raises the possibility that this can be behaving the way John Listman has hypothesized in his 1995 paper. What is that? Normally they exist in this closed state. When they exist in this closed state, then the active site, right, the site which is used by the enzyme to phosphorylate or to carry out its activity, you call that as active site, that state is not accessible for other molecules. As a result, you can think of that enzyme is existing in an inactive state. However, when it binds to calmolidin, or when you have a phosphorylation, what you have is there is a steric hindrance whereby the molecule by per se cannot close itself and letting the active site exposed. So, that now it can remain in an active state by itself. Now, this can also happen when it binds to some other protein, that can happen through another site. So, this can also be because of another calmolidin binding or some other protein binding there. So, the these are the key aspects of the calmolydin molecule that allows us to postulate probably calmolydin kinase Chem K kinase 2 molecule that allows us to postulate maybe these molecules are useful in preserving the memories, in maintaining their memories. So, summarize quickly, the molecule exists in two states, active state and inactive state, and going from inactive state to active state requires phosphorylation or binding to some other protein molecules of its own kind or some other kinds, and it can happen, the phosphorylation can happen by itself, meaning by another molecule of its own kind, right. So, that happens at this particular residual site, it is called the threonine 286 site. When it does, it goes into that active site, constant active site mode. So, now this is telling it is likely that these molecules can take part in encoding that memory and maintaining them, but the proof of the pudding comes when you actually can really prove without this molecule you cannot maintain the memory, and that is what we and guess what, we can actually do that, and people can do that, not now, as we can actually guess from the way we have progressed in the lectures. You can go and specifically take away this particular molecule and ask what happens to the memory, and that is what we will be doing in the next lecture, and show that really this molecule, why really this molecule is a good candidate and it is considered as a as a possible correlate of or possible substrate for memory storage. I will see you in the next lecture. Thank you.